Olá, pessoal. Boa tarde. Boa tarde a todos, a todas. Bem-vindos, bem-vindas a mais um momento da Semana da Engenharia Biomédica do Inatel. Esse ano de 2022 está bastante especial. A gente tem várias comemorações. Espero que vocês tenham conseguido acompanhar algumas delas. Inclusive, esse ano a gente tem os 10 anos do eHealth. A gente tem 2 palestras internacionais. Então, o negócio está bem movimentado por aqui. Agradeço a todos que estão nessa sala, no auditório. Agradeço também ao pessoal que está em casa, em qualquer lugar que vocês estejam assistindo pelo YouTube. E hoje, nesse exato momento, a gente vai ter mais um desses momentos muito especiais, que é conversar com a Dr. Kang-Mieler. Ela é especialista em engenharia biomédica, em nanopartículas, e ela trabalha na área de implantes oculares. Tem uma interseção muito próxima com a Emma, a Emily Dosmar, que conversou com a gente na segunda-feira. Ela também foi minha orientadora na época que eu fiz intercâmbio, foi orientadora da Emma no doutorado, e é uma pessoa fantástica, com uma bagagem de experiência incrível. E quem tiver alguma dúvida, por favor, pode acessar o link, ou quem não quiser acessar o link para mandar a sua dúvida, fique à vontade para chamar a turminha que está aí do lado, que aí depois, na hora das perguntas ao final, vocês podem direcionar para mim, que aí eu vou e repasso para ela, ou quem quiser perguntar também, pode ficar à vontade, tá bom? Então agora eu vou passar a palavra para ela, para a Dr. Kang-Mieler, e a gente já dá início à palavrinha dela aqui, tá bem? Hey, Dr. Kang-Mieler, now you just started. So again, thank you so much for coming. I'm very happy for having you here. As I said to you once, it was for me a pleasure for having had you as my advisor, and you motivated me a lot. I know your passion for science, and you have doing so many things very great. And I'm still working with nanomaterials and biomaterials because of you, so I'm very glad for having you here. Thank you so much.
Well, thank you. Well, it's been my pleasure. Thank you so much for your invitation, kind invitation. It was such a nice surprise to hear from you, and I'm very happy that you're in academics and pursuing your own career in academics. So it's actually my pleasure. Um, all right, so should we start it? Yeah,
Started,
yeah.
so the audience is ready for you. Thank you so much. I'm going to
All right,
pass the
may I?
slides for you, okay?
Yeah. I'll have my slides. All right, so may I have my slides? Perfect. Okay, so I'm gonna. Yes, I can see it, but there's still a little bit echo, so I think you need to turn off your microphone. Oh, okay. All right. Well, thank you, everyone. Like I said, thanks to Felipe. It's just really nice to be able to give you actually virtually, not in person. I've been to Brazil many times, and it's a beautiful country with wonderful people and culture. And it's really nice to hear that my former student is now a professor, and it's nice that the academics are continue growing. So again, thank you for your time today. I'm going to talk about today some of my work related to drug delivery system, particularly used for treating the retinal diseases. Next slide. So here's my financial disclosure. I do have a US patent on the system that I'm going to talk about, and I do have another patent pending on that. And I do also have research grants, particularly two NIH R1 grants to support some of my research areas. Next, please. All right, well, some may say that the eyes are the window to the soul. Well, next. Oh, it didn't work. All right, that's all right. There was a little animation. But if you really look at it, the eye is a really beautiful place to work. If you look inside the eye, this is kind of what you'll see. Unfortunately, the images sort of is hiding, but what you see is this beautiful optic disc, which is sort of the hole that you see in the back. And you see the blood vessels coming out, and then you'll be able to see that blood vessels going into the tissue, which is the retina. Now, the retina is a part of central nervous system. So retina is just like the neurons, is made up with different neurons as well as glial cells. And it's actually a beautiful place to work because this is really a unique place where we can examine the coupling of neuronal function with the vasculature non-invasively. So you can really look at how the vasculature plays a role in retinal function as well as vice versa. Next. Now, because of this inner play of neural functions and vasculature, is the really reason that I got into vision and ophthalmology. Particularly, I'm really interested in understanding the disease progressions and the treatment options for retinal vascular diseases, such as age-related macular degenerations or AMD, diabetic retinopathy or DR, or retinal vascular occlusion. Now, you don't have to be an expert to notice that these pictures, these fundus images are very much different than the image that you saw in previous slide. You can see that there are lots of blood, lots of hemorrhages in the eye, and a lot of the blood vessels looks unhealthy, different. And so these are all part of a disease process that actually occurs in retinal vascular diseases. And unfortunately, the consequence is a severe vision loss. Next. So my laboratory is really interested in translating some of the tools that we develop to really enhance the clinical treatment of retinal vascular diseases. These are the 4 areas that I'm interested: ocular drug-delivery system, retinal imaging and biomarkers, retinal hemodynamics, and also electrophysiology. But today, what I'm going to do is just really focus on one area, particularly in drug delivery system called STAR DDS. And this is what I call sustained treatment with anti-VEGF agents for retinal diseases drug delivery system. Next. All right, so here are some numbers for AMD, and we're going to really focus on AMD rather than other vascular diseases, okay? So AMD is #1 leading cause of vision loss in people over 50 in US, and that's just US. Think of it globally, the number is huge. There are 2 types of AMD. One's called dry AMD, which is much slow in progression. And there's something called wet AMD. And wet AMD is when we have a vasculature involvement. And the consequence of wet AMD is a severe vision loss. And that's where I'm really focused on. Now, 196 million people worldwide have AMD in 2020. And now this number is projected to increase to almost 300 million by 2040. So as you can see, the number of patients affected worldwide is huge. 50 plus is a typical group age that are susceptible to the AMD. And if you look at the global health cost wise, over $340 billion were directly linked to visual impairment due to wet AMD. And that includes about $200 billion of direct health costs. So when you think about this problem, this is a critical, really clinically significant problem. Next. So as I said, wet AMD is one other form of the AMD that actually results in severe vision loss. What happens in wet AMD? Well, it develops when the blood vessels grows under the macula. Now, the macula is an area in your eye where we use for central vision. So like how I can read, how I can see the colors. Now, when the blood vessels underneath that macula grows and interferes with the macular function, well, it becomes a problem. Their blood vessel can leak blood or other fluid to the back of the eye, and they can actually cause scars, and that causes symptoms such as blur lines, wavy lines, colors changes, or even blind spots. And here again, you can see the comparison between normal fundus and wet AMD, and you can see that it's very, very different than the normal fundus. Next. So what happens to wet AMD when you have it? Well, um, click once. So you can see, next, can you click on, yeah, I don't think, okay, actually, can you go back to one more slide? The animation is not working, so that's fine. So if you look at the top, the left box, you can see the half of it is a normal vision and other half is what AMD patient will see. So you can see that the images can get very, very blurry. You can see the straight line becomes very bendy or wavy. Some patients actually lose some sort of color perception. So instead of seeing this color, you might see these a very faded color. And others may see a shape sort of distorted, as you can see in the last box. And so these are all symptoms that the AMD patients kind of go through. And as the disease gets progressed more and more, it gets severe in terms of visual function. And eventually, a lot of patients actually do lose entire vision. Next. So what are some of the treatment options? And the options were actually very limited. So in 1973, thermal laser treatment was first proposed and used. And the idea of the thermal laser is essentially take the laser and destroy or seal off the blood vessels. So they aim to, shoot this laser in the eye and try to get these blood vessels destroyed. Now, the procedure produced many scars, and that was a really problem. The retinal scars will cause blind spots and in the visual fields. And like I said, as you get more and more further into disease progression, you're going to have to get a lot more of these laser treatment. And at the end, it really did not provide any use for vision or maintain the vision. Now, up until 2000, again, since 73, 2000, there was nothing else that you can do. There was some, procedure of surgical that can relieve some of the symptoms, but no treatment. Now, in 2000, another treatment was proposed. It's called photodynamic therapy, which is called PDT, which allows Visiodyn, which is essentially a dye, and using low energy laser to, again, try to destroy these blood vessels. Now, with this low energy level of laser, with the combination of dye, they were much better in targeting blood vessels than just hitting the blood vessels. But 1 in 6 patients showed the improved vision, but really it's still not an effective, and it was really minimal use. Next. So back in the 1980s, there's been studies shown that the vascular endothelial growth factor, VEGF, are elevated in wet AMD. Now, we know that VEGF in the 80s, when it was first discovered, we knew that it was involved in vascular or neovascularizations, and subsequent studies have shown that the levels of VEGF are much, much higher in wet AMD than control eyes. Next. So in 2004 was really the first anti-VEGF agent was FDA approved. So what that means that these drugs were actually blocking VEGF production. So then actually we then kind of into anti-VEGF therapy era. Next. What's that mean? Well, now patients with wet AMD are getting these anti-VEGF injection in the eye. So classical clinical studies such as Anchor or Marina demonstrated for the first time monthly intravitreal injections of these anti-VEGF, in this case ranibizumab, result in a significant vision in AMD patient. Now, this is a really exciting and really exciting news, because not only that you're slowing down the progression of the visual loss, but the patients are actually gaining back the vision they have lost due to the disease. So this was a really exciting time where when we were finally able to offer some patients a potential treatment to maintain their vision. Next. But here's the catch. So even though the anti-VEGF therapy really changed the way we manage wet AMD, the therapy still requires a direct injection into the eye. And so this is a picture of an intravitreal injection. And so think about getting these injections monthly for a period of time. It's not a 1 month, 2 months injections. Some of these patients have lifelong treatment is needed. Next. So then why do we do intravitreal injections? Well, if you look at the eye is actually a very interesting place. You can actually deliver drug in many different ways. First, there's a topical delivery, so the eye drops that we all see. These are one way to do it. There's a transcranial delivery and subconjunctival delivery, and also intravitreal injection, which is going right into the vitreous. But the intravitreal injection really is the most efficient way to get the delivery to the posterior segment. Now, posterior segment, what I mean by is to the retina. That's where the problem is. Now, you might say, well, why not do the topical? Well, the problem is that some of these anti-VEGF are large protein biologic agents. They're too big for topical to be delivered topically. The agents will not penetrate into the cornea and other tissues and get to the back of the eye. So I'm not sure why they kept showing my former graduate student's name there, but that's not my name. Anyway, but what's really also, even though this is the most direct way to get into vitreous, there are some potential complications. And ophthalmitis is really one of the complications, which is infection in the eye. They could have retina tear or detachment, so the retina from the back of it can actually come off. Cataract formations due to multiple injections, you can actually damage the lens and can have cataract injections. But really, one of the really interesting and also another problem is the bolus amount of drug is delivered. Because you got to deliver this high load of drug into the eye so they can last for a while, you need to actually give a large concentration at given or every month. Next. So what's the limit to intravitreal injection? Like I said, one of the things I said was a bolus injection. So imagine if you get one injection first month, your drug concentration in the eye is very, very high. But the anti-VEGF have a very short half-life, and because it's going to clear out of the eye by these retinal circulations and other ocular circulations, the drug will quickly leave the eye. So then you have a very low amount of drug. So then you have to inject again to get the high amount of drug. So you have this roller coaster of the drug concentration that's exposed to the eye. So you could have potentially too high of drug concentration, could potentially be toxic, or you could have these low amounts of drug, which is not maybe very effective. So what we really need is a constant sort of steady level of drug delivered throughout the entire treatment period to really have a beneficial, and that's where the drug delivery system comes in. Next. So all anti-VEGF therapy, even though they're very effective, have a common limitation. So in order to be effective, a monthly injection is required. Okay, so think about that. Every month you have to go to doctor's office and get these injections done. So even though the anti-VEGF is very, very effective, the burden of a monthly or even bi-monthly visits and injections have on the patients, their families, the physicians, and even our healthcare system cannot be denied. So we need a better solution. Next. So that's where the drug delivery system of anti-VEGF comes in. So up until this now, there really wasn't any FDA-approved drug delivery system for the eye, particularly delivering anti-VEGF. Now, in October 2021, FDA finally approved the first device that can deliver ranibizumab, which is another anti-VEGF that was made by Genentech, for the wet AMD, and it's called port delivery system. Now, this is a non-degradable, refillable, surgically implanted device. And so, idea is that this non-degradable implant gets in the eye, and the way it was designed, it actually exposes from the eye, so you have access from the outside, and you can actually refill these ports every 6 months. Now, it's exciting. I think this is really one of the first entry into sustained delivery system for the ophthalmology. It's been effective, but yet there is some issues. Since then, I mean, we've seen some cases of complication with either surgical procedure of it or maintaining in the eye for a long period of time, where the the implant has caused infections and complications. But still, very exciting time, very exciting that the first implant was actually used for sustained delivery. Currently, there are no FDA-approved biodegradable device that can really deliver anti-VEGF or in a sustained manner. The reason is these antibody-based anti-VEGF are really large in molecular weight. We're talking, you can see in here anywhere from like it's 25 to 100 and some kilodalton, which is actually huge molecules. They're very delicate in nature. As you know, that working with the proteins, it's always challenged to keep the bioactivity, and also how much is available to the tissue, to the eye, has always been very challenging. Next. So there really is a great clinical need for extended release that is biodegradable ocular drug delivery system. Next. So what would be the ideal drug delivery platform? So the platform should really have some of these following criteria. To be delivered via relatively non-invasive method. Granted, it would be great if you can do this topically, but sometimes it's very hard to achieve the drug concentration. But if it's a very non-invasive method, that would be favorable. You should have a really good release profiles and have a profile that are sustained, not one of these very all-in-one release. And offer really high encapsulation efficiency, which means that you want to have a lot of drug in the device so you have be able to deliver for a long period of time. And it'd be something that can be easily manufactured and used and store for the clinical use. Next. So this is where we come in. We have developed a biodegradable system called STAR-DDS, and I'm going to call it just STAR-DDS or our drug delivery system from this point on. And this is, I'm going to just show you what we've done to sort of try to address this clinical problem. Next. All right, so STAR-DDS is a composite system. So we're using PLGA microspheres that will encapsulate the anti-VEGF, whether we're talking about the ranibizumab or aflibercept or any other anti-VEGF for extended delivery. And in fact, actually in this talk, I'm going to really only focus on delivery of the aflibercept, which is a type of anti-VEGF that's been commonly is one of the most popular anti-VEGFs be used. But our system can work for other type of anti-VEGF. And what happens is that once we load the drug into the microspheres, this drug-loaded microspheres that gets then embedded into thermal-responsive polynipam based hydrogel. Now, the reason that we put this into hydrogel is for several things. It really confines the microspheres to the delivery site. So that the microspheres, once we inject into the eye, microspheres will stay within the injected area. Others have tried just injecting microspheres directly into the eye, and they cause lots of problems, ranging from inflammations to microspheres, floating around and migrating and escaping. And what's nice about the thermal-responsive hydrogel is it's a thermal-responsive, it's sensitive to temperature, and we can control the transition temperature within our polymers. So our system is that at room temperature, it's what I call more like a liquid-like gel that you can actually inject through a small gauge needle that we use the same size needles in humans and gets injected into the eye. And once it gets injected to the eye and exposed to body temperature, the polymer collapses and becomes a solid gel and stays in place. So what we've done is that we're using same intravitreal injection procedure, which is now we've done many, many times that we are very, very comfortable with. So our clinicians is not changing the behavior or procedure. And it's the same procedure. And with the same amount of volume that we inject, we can actually achieve a similar drug efficacy. Next. So here's our thermoresponsive hydrogel and why we've chosen it. Like I said, it is temperature sensitive. So at a room temperature, it's well hydrated chain. And as soon as we increase the temperature, it collapses by losing the water. So, and we can control that transition temperature, what I call here volume phase transition temperature, depending on the cross-link density and so forth. But typically, we aim to be about 32 degrees. So at around 32 degrees, the transition from the liquid to solid is very fast. So within few seconds that we inject, the gel does collapse very rapidly in the eye and able to release the drug. Next. So first, one of the first thing we've done is to look at how well can we actually control releasing our drug. So like as remember, I'm going to just talk about the aflibercept as an example. So we first looked at the in vitro release samples and see what the profile looks like. So the aflibercept was radio labeled with I-125 and incorporated into microspheres. And that microspheres then gets embedded into hydrogel, just like how we usually do in our manufacturing. And so, but then what we did is the drug delivery system, start DDS, was maintained at 37 in a buffer. And at predetermined time, we would then collect the samples and determine the radioactivity of the sample. Yeah, here we are defining initial bursts as a drug release within the first 24 hours, and drug encapsulation efficiency is a percent drug within the sample of microspheres relative to theoretical loading. So as you can see here that on the table, we got about 53 over 50% encapsulation efficiency for the drug getting into microsphere, which is pretty good. Typically, I think if you can hit about 30% or above, you're doing pretty well in terms of these manufacturing of microspheres. And by the way, we're using double emulsion technique to do so. So then within that, we got over about 75 or so, between 70% or so of the microsphere gets embedded into hydrogels. Next. So here's a release profile. So, and we're looking at two different types. One has only about 10 micrograms of microspheres loaded into gel, and other has a double the concentration. So as you can see, it nicely doubles the amount the drug that we can load into the gels as by introducing more microspheres into the hydrogel. Our drug loading is anywhere from like 100 micrograms to close to 200 or so. We have initial burst about 37 or 47, depending on which formulation you're using. But what's really nice about it, after the initial burst, there is actually really controlled release on the range about 0.11 microgram per day, which is actually really nicely maintained for over 200 days. So now over 6 months period of time, we could really achieve this very nice, steady, a small amount of anti-VEGF can be released to the eye that would be effective. Now, somebody asked, well, what about this initial burst? Is that too big? In my opinion, actually, it really is not a big concern for us. We think of that as sort of almost like a loading dose. You want to give a big dose of a drug first and then maintain the concentration. So we don't see the initial burst that we see as really the big concern. Next. And here's just another way of looking at the release of aflibercept and also seeing the bioactivity of these release sample. So sure, we're really happy that we can maintain for 200 days, but if we're releasing the bioactivity, releasing the protein that are not bioactive, it really is not very useful. So here, this is a plot of bioactive aflibercept was released in these in vitro studies, and we utilize it to quantify and examine the bioactivity. And as you can see here, the release, you'll see the big sort of the initial amount of release and then sort of a steady level towards the durations. And what you see is that the reported number is much higher than the aflibercept binding affinity concentration or as well as IC50. So we know that even though we're releasing small amounts, it is still very much bioactive and it is still higher than what you need for IC50. Next. So then we also looked at the potential cytotoxicity of a STAR DDS in vitro. So using cell culture system, HUVEX, we test to see whether One, if the system is toxic to the cells, and also what happens when the byproduct of the degradation of the samples and how that might be actually harmful to the cell. So what you see in the gray bar is that what in our practice, when we actually make this star DDS, is that we actually wash our star Ds with a large volume buffer for several reasons. One, we want to make sure that we're actually cleaning these, and because eventually these will be also injected into the humans or animals. But second thing is we want to actually get rid of a lot of unreacted polymers that may be present in the surface of the star DDS. And indeed, the first two washings, you can see that there are, it's affected by cell availability testing. You can see that there is some effect on that. So we know that we're really needed to make sure that the sample has been washed properly. But by the wash 3, 4, and 5, really there's no changes, and we know that it's actually quite safe to inject. The dark bar is showing the samples, degradation samples over time, and what we did is again collect them, expose to the cells, and see if there's any changes in the cell viability. And as you can see, there really isn't any effect by the degradation byproducts that has effect on the cells. So we're pretty confident at this point that our system, our procedure can produce a very stable star DDS with the ability to be injected. And so we are now moving on to an animal study. Next. So what we did, so I'm not going to go all the different types of animal studies we've done, but I'm going to kind of highlight some of the things that we have done and where we're going. So first, we looked at the CMB animal study. So CMB, which is a laser-induced choroidal neovascularization, it's an animal model of wet AMD in human, in a simple term. So we can induce these lesions in the back of the eye, just like a humans would develop, and then we can then treat the animals with our star DDS and see whether it's effective in terms of reversing some of these CMB lesions, as well as also testing how safe it is to inject into the eye. Next. So first, we started with the rodent models. So small animal models, but these are well-used, well-characterized rodent CMB models. And so what we've done is that we use an imaging tool called fluorescein angiogram, or FA. And so that essentially injecting dye into intravenously, it circulates into the eye. We can see these beautiful images. On top image, you can see blood vessels, and also you can see the lesions that gets highlighted because the blood vessels that were created by these lesions are also showing the leakage of the dye. So what we did was that we induced laser inductions of CMV. We waited 2 weeks. We waited 2 weeks to ensure that the lesions are fully developed. And then the animals are divided into different groups. So the group 1 had no treatment, so it was a control group. Second group is a positive control group where it had lesions, but it also treated with a blank DDS, blank star DDS, meaning there was no active drug in it. Third group was what I call clinical group. So we're injecting bi-monthly aflibercept. So if I was a patient, I would get these injections bi-monthly, particularly for aflibercept. And so each injection will give 200 micrograms of aflibercept into the eye. Now, fourth group is one injection of star DDS that is loaded with aflibercept. And in this group, with one injection, you're only getting 1 microgram of the disease or of the drug. We then follow the animals multiple times throughout our study period, and we take these images. The same animal can be used over and over because these imaging are non-invasive. And then so we can actually see the changes in lesion size over time. And at the end of 6 months, we then sacrifice the animal and get a histopathology. Next. All right, so how do we quantify CMV lesions? So the previous works that people have done is that they had to sacrifice the animal and do histology to measure the lesion area. And the lesion area I'm talking about is these round spots that you see around in between the blood vessels. Okay. And so obviously that's not going to really be all that helpful when we do longitudinal study because we really want to see if the same lesion kit gets smaller and smaller over time. So we developed this imaging method called the multi-exit threshold method, MOT, where we're taking the fluorescent angiogram lesion image and then we're running through these very simplified imaging process to measure the actual pixel size is occupy the lesion and then convert it into area. Next. All right, so this is just a representative FA images from the animals of 4 different groups. As you can see, no treatment, blank DDS, bolus injections, and star DDS injection. Week 0 means like right after we treat it, and then week 22. And qualitatively, you can start to see kind of the area, the lesions getting smaller and smaller on the later week. But let's look at more quantitatively. Next slide. Here is a box plot showing the same 4 groups at a different time points, as you can see. And you can see that the positive number is the lesion area are getting bigger. And if you go see the negative number, the lesion is getting smaller and smaller, so eyes are getting better. Now, let's look at the no treatment or blank DDS, which is dark red and orange. As you can see, entire time, the lesions do not regress. They're either staying about the same size, or in fact, some animals actually got even bigger. So what that means is that since there was no treatment, no drugs, these lesions did not get better, and it just sort of stayed the same for the entire the period. But now let's look at the greens. The light green, which is a sort of called clinical multiple injection of aflibercept, and the dark green is our 1 injection of star DDS. And you can see that the lesion size start to regress, gets smaller, and it is actually quite significantly different than the controls. So we were actually very excited that the lesions are actually regressing. But what's really exciting about this is that our star DDS group only got 1 injection, which is only totaling about a 1 microgram of the aflibercept, where these bi-monthly bolus group got 3 injections, totaling 600 micrograms of drug. So what's really interesting and really exciting for us is that 1 injection with much lower dose that was released for 6 months had really about the same effect, if not even better, than the so-called clinical group where they got multiple injections. Next. And here is another confirmation. At the end, we sacrificed the animal and got histology. Again, same thing as we've seen with the MOT analysis. And histology shows that the treated animals have a much smaller lesions in histology. And in fact, they're pretty comparable. Again, sort of confirming our imaging analysis that we've done to really show that there is an effect of our drug delivery system. Next. So this, I'm not going to go in a lot of details, but what's really nice about our models, our lab, is that we're actually able to do not only looking at the imaging, but also the cellular function. Remember how we said the retina is sort of like a neural size and actually will, you can actually measure the cellular function. So ERG is sort of like, think of it as like an EEG or EKG, measures the retinal cell functions. And so we're using this non-invasive ERG technique to see whether retinal cells are staying healthy throughout the entire time. And the MB waves are the different components that made up ERG that reflects the activities of different cells in the retina. And so there was a small transit changes in the overall amplitude of the A wave, as you can see in the top left corner, but really the change was much smaller in over, remember these are global function, not sort of an individual function. And if you look at, but what's really interesting is that when you look at the sensitivity, so meaning that if you were to measure the retina exposed to a lot of different lights of the light spectrum and see the overall effects, there really isn't any changes between the pre and post treatment. So overall, the retinal cells remain very healthy, normal after the STAR DDS injections. Next. And we also looked at the intraocular pressure. So what happens with in animals as well as in humans is that when we actually inject, the pressure inside the eye could change. In fact, that's actually really bad. If the pressure gets really, really high and doesn't recover, then you will actually have other type of complication. So here we measure the IOP. Again, we can do this non-invasively before, right after injections, and as well as following the entire time study period. So you can see it, not surprisingly, when we inject the our SAR DDAs, there was a spike in the IOP, and then it eventually recovered. But there's two things I like to point out: that the spike and increase, even though it's about 28 millimeter mercury, is still within the normal IOP range of the rat. And also, what's really nice that it eventually did recover and maintain similar to that of the pre-injections. Next. And so I'm not going to go too much in here, but we did also a lot of clinical exams of these animals, both not only inside what's happening, but also outside. Just so you can look at visually, there's really no changes in the ocular. There's no obvious infections or things that could actually affect the vision. The lens and cornea will remain really clear the entire 6 months. Again, no inflammatory or infections that we're observing. And you can almost see at the bottom row, that big blob that you see, those are actually the STAR DDS. And so you can see over time, it's getting smaller and smaller as it gets to degrade. So it's a kind of nice picture that you can see in vivo image of your biomaterial degrading. Next. And this is another histopath looking at after 6 months. Basically, there was no sign of chronic inflammation or foreign body reaction in both the front of the eye or the back of the eye, which is great news. One of the biggest concern and one of the biggest concern that other drug delivery system that are in development right now is the chronic inflammation. And that's a really big problem. You don't want to trade one problem for another. So at least in our rodent models, we have not seen any inflammations. Next. So, based on this really exciting small animal work, what we are very much encouraged by the fact that our STAR DDS can be very effective. It could be potentially a option for clinical trials, but obviously we can't jump right in from the road into the clinical trial. So, last couple of years, what we've been is to working with a non-human primate model to really demonstrating the fact that all our things that we've done in the rodent models are translatable to non-human models, and hope that based on that information, we'll be able to go to FDA for FDA approval to be used in humans. Next. So I'm going to just in the next few minutes, we're going to talk about the non-human primate studies. So one of the first studies we did is a short-term safety, and the safety is going to be one of the number one thing the FDA is going to ask, and how safe is it to inject and have not a problem. So we took a group of macaques, and one eye got injection of STAR DDS, and other served as control, which means it had no injection. In our primate studies, volume that we inject is 50 microliters, which would be the same volume that we would use for humans. And in fact, we use these monkeys because they have about the same size as human eyes. So the information we get from these primate studies really are beneficial in understanding what would happen in humans. So what we did in the short-term study is that we looked at monthly we did one injection, we did monthly exams every month for 7 months to see if there are any safety concerns that we can find. And then at the end, we did a histopath again to prove that there was really no concerns. Like the rat model, the STAR DDS that we use, and this is very, very similar to that. The microspheres degrade within about 6 months, and the hydrogel degrades about 7 to 8 months. And this is done by design. And the idea is that the microspheres are encapsulated into hydrogel. It degrades and releases the drugs. And once it's done, then the hydrogel, which degrades much slower rate, will start to degrade and disappear. Again, the idea is to make sure that we keep the microspheres in one area so we know that they don't migrate to other tissue. Next. Okay, so here are some of the external and fundus images of the primates from the baseline to 1, 3, and 6 months after the injection. Again, you don't have to be really an ophthalmologist, but look at it. There's really no really obvious signs of inflammations or changes in the eye, both external and internal. In fact, I have a collaborator who's actually helping with this study, and she is actually a veterinary ophthalmologist, and she's the one who actually performed all these clinical exams for me. Next. All right, so one of the safety studies, so we're going to kind of go down the line of safety. So we looked at cornea. Are there any changes in cornea, which is the clear tissue in front of the eye? If that gets messed up, you're not going to see a good vision. So we looked at different types of the clarity of the cornea. We looked at the cell density, axial length of the cornea, and as you can see, that they are pretty comparable. So the black symbols are the treated eyes with one injection of STAR DDS, and the red are untreated controls. And all the parameters you see are pretty much on equal to or similar to that of control. Similar to the rat, we also looked at intraocular pressure. Again, remember, you don't want the pressure to go really, really high after injection, because then, one, you'll feel the pain, and you can actually have consequence of effects on other cells in the eye. And as you can see, that there really wasn't much change at all in terms of intraocular pressure from the injection itself. And again, we're injecting about the same volume that you would inject for humans. So we are very, very confident that within our injection in this parameter, we're not going to have any IOP issues in humans. Next. All right, so then we also look at the retinal thickness. So the ophthalmologist actually has lots of fun imaging tools. Another tool is called OCT, which you can see the images on the left. Essentially, what I call OCT is like living histology. You are literally looking at this cross-section of the retina and looking at the different layers of the retina and the choroid to see the structural changes. And it's a fantastic way to actually make sure that nothing is actually changed within the retina. And we measure the different thickness of the retina to make sure that there is no changes. And I don't have a picture of what would happen if you have a problems, but basically we measured a lot of different areas of different thicknesses, different location of the retina, and making sure there's no changes. And again, if you look at the, comparing the control versus the injected eyes, really there was really hardly any changes at all in the retina thickness, which is a great sign that our, the tissue is maintained. Next. And we did, similar to a rat study, we did an ERG. So again, ERG is actually a clinical tool. So, you get this done in clinics all the time. So we did the same thing for the rats, same thing for the humans. And once again, looking at the different parameters you can measure with the ERGs, really there's hardly any changes at all between the control eyes and the injected eyes. So this is actually a very, very good sign that their cellular function is intact and working normal. Next. So, and this is a histopath at month 7. So after 7 months later, we sacrificed the animal, got the tissue, and looked at, if there's any effects on that. The arrow they're pointing to in the eye, which is sort of right behind the lens, is just the leftover STAR DDS. So there are several things that were very, very encouraging. There was no evidence of a migration to the anterior chamber, so meaning that once we inject, it stayed there, it didn't move around the eye at all. Second, it was really well delineated, and there was a very thin fibrous layer around this fiber, and that around the STAR DDS that consists of perhaps a small foreign body reaction. But what's really good about it is that it's not an inflammatory reaction. And we know that this is the same animal we observed monthly for 7 months, and this is a result of the histology. There's no inflammations, and the small body reaction did not have any effects on the functional of the retinal cells or even the structure of the retina. So that was a very encouraging short-term safety study. Next. So based on that, what we're doing now is we're doing long-term safety study. So a subset of animals that we have observed got a second injection into the same eye. And now we're doing, again, monitoring with the same parameters that we just talked about. And where our goal is to be able to monitor these animals for up to about 2 years endpoint. We're about almost another year into it now, year and a half. And so we're very excited to see what happens. So far, the animals are doing great. No obvious signs of inflammations, no obvious signs of rejection of the STAR DDS injections, no obvious signs of changes in cellular functions or even structural functions, both externally and internally. So we're very much encouraged by our long-term safety study. Next. And another thing we are now trying to do, and we are in process doing it, is doing the CMV study. So remember I talked about the CMV model that's for the rodent, the rat models? Well, we can do the same thing in monkeys. So we can actually induce these lesions, CMV lesions in the monkeys and see if it can actually make it better, just like what we did with the rat models. So we are in process doing this. Again, the procedure is that here's sort of initial data we saw is that we induced the CMV, waited 2 weeks, treated, and then follow with FA, fluorescein angiogram to see whether the lesion size changes. Next. And so what you see qualitatively, the left is a week 0, so right after you treated the animals, the 2 weeks and the 4 weeks after the treatment. Now, qualitatively, the lesion looks better. It's a lot less leaky, so it's not as brightly, fluorescent as you see in the week 0. And then we use MLT to see if we can measure the area. Next. And what we see indeed is that the area, got bigger, but then after treatment, it starts to regress. So on average, by 4 weeks, the CMV lesions were regressing to near the baseline or near the pre-treatment. So that was encouraging. But then we actually had a little bit of a roadblock, if you will. That CMV lesion that was in the control eyes start to regress in a month 2 without a treatment. So this actually kind of complicates our interpretation of our STAR DDS. Next. And so we think there's 2 things. So we saw in the literature that the primates, unlike the rodent models, sometimes do not will have a spontaneous regression of CMV much at higher rate than the rodent models. So sometimes without a treatment, it just gets better. So that's one thing that we noticed. So we are now really selectively selecting animals and selectively making sure that our lesions are well placed in the right place with the right amount of laser to create a perfect lesions. Second thing is we noticed that the MLT analysis that we have used actually was really optimized for the rodent FA images, not for primates. And so it could be that the primate lesions are leaking a lot more dye and we're actually missing some of these changes because we're not capturing with our MLT analysis. And I know I didn't go in a lot of details why that is, but just the way we analyze it, perhaps we're missing it. So now we're going back and reanalyzing all our images, and by using sort of a semi-qualitative grading system. This was published a couple years ago. We can look at the different size and different amount of the dye that comes out of the blood vessels, and we can kind of, assign 1 to 4 different types of grading to kind of characterize it. It's not perfectly qualitative, but it does kind of give us a way to compare different lesions at different time points. So currently we are in that reanalyzing some of our images, and we actually have animals now going out to months beyond the second or 2 months. So we'll see what happens. I think this so far looks promising, but stay tuned. All right, next slides. I think we're almost there, guys. Okay, so we have, our primate studies are still ongoing. We have the long-term safety study I just talked about that we're going to go out to at least 2 years to see if we can establish the really good safety profiles for STAR DDS. We are reanalyzing our wet AMD, the CMV images again, just with our using qualitative, semi-qualitative grading system to make sure that we can see the treatment efficacy. I think we should be able to demonstrate with the using that instead of using the MOT analysis that we did for the rodent. And we are also doing intraocular pharmacokinetic studies. We have 7 months PK studies out. We're just now waiting to read the getting the last bit of the samples to be analyzed. Very soon, we should have a really profile of the intraocular pharmacokinetics of the SR-DDS in primates. Next slide. Okay, so for this, just a summary, our STAR-DD is really the first biodegradable system to release bioactive anti-VEGF for over 6 months. So idea is that patient will get 2 injections a year, every 6 months or so, instead of these monthly or bi-monthly injections. We see that the STAR-DDSS actually really have superior treatment efficacy with a lower overall dose, particularly in rodent models. We see that with 1 injection with much lower dose, we have treatment efficacy, and we're hoping to see that in primates as well. It has excellent safety profiles, as you saw in both rodent models as well as the primate models. Both cellular functions, structural, vasculature, they were not compromised by the SR-DDS. And our treatment, if you go to every 6 months, it will really reduce the treatment burden on the patients and physicians in our healthcare. So we do think that STAR-DD is this novel system to replace and improve the current wet AMD treatment. When we'll be ready, hopefully soon, but we still have a lot of work to do. But we're very encouraged by our data that we've gotten so far from both small and large animals. And so hopefully in short near future, we'll have something like this system to be tested in humans. Next slide. And I just want to thank all the team. This is not just one person work. I have lots of folks in my laboratory, my collaboration at UIC, UC Davis, and now I have a collaboration with the University of Wisconsin-Madison doing histology. So there are lots of people involved in the STAR-DDS that I'd like to thank them. And that is all. So if you have any questions, I'd be happy to answer. Thank you.
Hey, Dr. Kang-Mieler, thank you so much for your presentation. Oh, can you hear me now? Yep.
Yeah, I can hear you. I can hear you.
Oh, great. It was a great presentation. I've seen so many photos and data I've seen when I was working with you in the lab, and I'm very happy you got so amazing details and results with the animals and models you've been working on. Thank you so much for your presentation. And I also have some questions here for you.
Sure.
Some of those questions the guys just sent while you were presenting, so maybe you had response, but let me see here. First question is, if the person has flu, or in this case, the animal has flu or cold, would that affect the performance of STAR-DDS?
No, because we're talking about local temperature in the eye. Unless your body temperature increases so high that you start to affect the interior part of the eye, that could be a problem. But in most cases, if someone has flu or cold and has increased temperature of even up to 100, 103, it's not going to really impact the system. And in fact, if you have higher, remember that our system is a higher temperature, the system becomes more solid. So so you would actually stay much solid, more solid piece rather than become more fluid-like.
Okay, thank you. Just an instruction for you guys here. If you have any question, just raise your hand and I can give your question to her. Translating to Portuguese. Se vocês tiverem qualquer dúvida, só levantar a mão que eu passo para ela, tá bom? And another question, Dr. Kang-Mieler, did you find any problem with reabsorption of hydrogel and spheres, like any toxic behavior or a limit supported by the retina?
So far, we have not seen any absorption by the tissue. So, longest we actually, I haven't shown here, but the longest time that we gone out with the rodent models, particularly, are over 2 years. So we had animals with injections, either single injections or repeated injections with the STAR DDS, and see if there are any changes in the behaviors, tissues, local tissue changes, and we have not seen any of that and any absorptions of that. So we don't think that's going to be a problem at all. In our primate studies, again, we're about a year plus out, and we still haven't really seen much changes because of the repeated or the injection because of hydrogel. So yeah, no.
Okay, thank you. Next question, which size of sphere is considered the best? They mean the bigger, the higher amount of drugs you can deliver, yeah. But they think in this question, if you put a big size of sphere, this might interfere in the injection. So do you have a problem with the size of the spheres?
So our microspheres are on average about 7 microns. So they're fairly small. Um, and it is true. If you go to bigger microspheres or particles, eventually you're going to have a hard time injecting into the eye because it's just too much bigger to do it. And so we were favoring more smaller, even though we are sacrificing the load of the drug that we can, get into microspheres. The reason is that we don't want to change the clinical practice. So our clinicians are very comfortable with the intravitreal injections. They know they could do it. They could do an office setting with really minimal, changes. And we really want to keep that practice same. And so we really wanted to make an injection where using small gauge needle. That's important because you don't want to start poking your eye with a huge needle. And then two, you want to make sure that the injecting is not as hard as if you're to change with the different materials. That's another reason we kept them small.
Okay, thank you. Um, I have another question here, just came to me. Thank you, Bruce. What's the expected time to move from primate testing to human one, and eventually treating even patients?
So here in the process is that so once we got our primate data, I think and demonstrate safety. That's really time that we can go to FDA and get an initial sort of investigational drug approval, which means that even though it's not fully FDA approved, we have a permission to use it to do clinical trial. That time could take a few years. We're right now in the process of finishing up our primate studies. We need to probably do one set of GLP, the Good Laboratory Practice Run Lab, to do the sort of the safety study again, just to make sure that we are following the guidelines of FDA. And have a meeting with FDA. So we're talking like in a few years that we can possibly have a meeting with FDA if everything goes well and our animal study has shown the really positive results. Once the FDA does give us approval to start a small clinical study, that's when we can start injecting and then do safety studies in humans. So we're still a few years out, but we're very hopeful.
Thank you. No. And the last question I have here, Dr. Kang-Mieler, is about magnetic particles. Do you know how the magnetic particles work, and does it have any applications for STAR DDS?
So the magnetic particles it's interesting. So my husband is actually a retinal surgeon. So it's nice that he and I can talk about a lot of different projects together. Actually, he's on part of my our team, obviously. So in the eye, actually, magnetics have been used to retrieve actually like foreign bodies. So like I said, my husband does a lot of surgery. So if you have injury and you have metal stuck in it, you can get the particle, magnetic to get him out. And so it does have some interesting properties. And you can make the particles sort of magnetic. So kind of idea is you can direct it and you can do target it into the localized delivery site. So there is some potential for it. And we gotta play with the idea of is that going to be really beneficial. The problem is that in order to really fully direct to the where you want to go, it's not as easy as it sounds. And so I think that is really one of the challenges I see with magnetic particles, particularly in the eye, because even though you can see it and you can kind of direct it, but if you think of it this whole sphere and you want to make sure you're not going to get into front of other parts and making sure that you're not going to interfere with the vision, you want to, it's going to be kind of hard to localize them. So I think that might be the challenge that you would have to, overcome. So yeah, it's an interesting idea, but I don't not sure how impactful it's going to be for in terms of delivery.
Okay, thank you, Dr. Kang-Mieler. Those
Mhm,
were the
mhm.
questions. So, in name of Inatel, the college I work for, and in name of the people and students here, I'd like to thank you very much for your time. It was an splendid time and moment with you, and thank you so much for your knowledge shared here this afternoon with us. Thank you so much.
Well, thank you. I really enjoyed the time. I wish I was there in person, perhaps some other time.
Yes, yes, I hope so.
Okay.
See you soon and keep in touch. Bye bye. Então, pessoal, para vocês que estão aqui, o pessoal que está acompanhando a gente pela internet, a lista de chamada acho que já apareceu aqui algumas vezes, né? Apareceu o link ali para vocês. Espero que vocês já tenham conseguido assinar. A gente também agora vai ter o nosso famoso sorteio, correto, Bruce? A gente vai rodar o sorteio aqui agora. Ah, claro, tem. Eu só vou. Então, pessoal que está na internet, espera só um momentinho que a gente vai rodar uma apresentação que vocês poderem ver. Ah, não estou sem o coisa, peraí. E para quem tiver qualquer dúvida, fique à vontade para perguntar. Segura pra mim, por favor. Agora é a hora que vocês dançam, né? Marcos, pode compartilhar se quiser, não sei se está aparecendo aí. O meu biscoito está aparecendo aqui. Muito bem, pessoal. Então, agora só para a gente poder encerrar esse momento, queria agradecer muito a vocês por estarem participando. A SEB é feita de vocês. Muita coisa legal aconteceu por aqui. Muitos momentos que vocês proporcionaram com as suas dúvidas e a participação. Então, muito obrigado. Gostaria de agradecer também aos patrocinadores, porque vários desses prêmios que vocês receberam ali na lojinha da SEB, eu acho que muitos prêmios já foram prêmios bons, então muita gente vai sair de barriga cheia desses momentos aqui, porque tinha uns prêmios que davam para fazer uma boa festa. E graças aos patrocinadores, então, EquipaCare, Master Medical e Biotron, muito obrigado pelo incentivo de vocês. É muito importante, são muito importantes esses momentos para a formação dos alunos e da comunidade do Inatel. Gostaria, antes de entrar nessa questão dos biocoins, agradecer muito, até aproveitando quem está aí online, a Maia Abramovic, que foi sensacional fazendo a tradução simultânea em português. A Maia é aluna do curso de Engenharia Biomédica. Maia, eu sei que você está por algum cantinho aí. Muito obrigado pela sua disponibilidade e participação, você é incrível. Agradecer também todo o pessoal da comissão aqui, Bruce, Marcos, Breno que está ali, João que estava no outro canto, os meninos que passaram a lista. Cara, vocês são sensacionais, têm feito um trabalho gigante. Muito obrigado mesmo. Aqui, lembrando então, né, a gente tem como ganhar os biocoins, para quem ainda não sabe, as aulas, webinars, workshop, palestra, passo ou repassa, que acontece na hora do intervalo, valem 3 biocoins. Nós também temos palestra de encerramento, que hoje acontece, hoje não, hoje é quarta, né? Hoje é quarta. Tô perdido no tempo, gente. Hoje às 8 acontece a palestra de encerramento, vale 6 biocoins e minicursos 9 biocoins. Pessoal, essa aqui é a lista atualizada até esse momento daquilo que vocês ainda podem trocar na lojinha da SEB. Oi? Oi, já tinha sumido? Pode voltar quem sabe lá repôs. Eu banco se não tiver lá. Brincadeira, banco não. Então tem alguns itens lá para quem ainda não pegou. Talvez alguns já tenham acabado entre um momento e outro, mas alguns deles devem estar por lá ainda, tá bom? Eh, Deixa eu ver aqui o que mais. E a loja da SEB, pessoal. A grife, né, da SEB, a grife da Engenharia Biomédica em geral, ele tem vários itens de roupa, caneca, chaveiro, uma série de coisas. Então, o arroba do Instagram é arroba grife underline engebiomédica. Vocês também conseguem mais informações ali na lojinha da SEB. Tem coisa muito legal, essa coleção que ficou bem bonita. Esses chaveirinhos, essas canecas aí, eu acho que vou comprar uns 37 só pra mim. Então, vale a pena vocês adquirirem os itens de vocês aí, tá bom? Agora eu sorteio. Falei, agora eu sorteio. Vamos lá, pessoal. Então vamos ver o sorteio. Link aqui na tela. Não sei se vocês ainda estão disponíveis para a gente, pro pessoal poder colocar. Quem não colocou ainda, então vocês têm mais 2 segundos e meio para poder acessar o link. Abro. Posso fazer o meu mesmo?
Gente, só para avisar, a gente tem duas pessoas participando. Não, 22? A gente tem 22 pessoas participando do sorteio, tá? Já encerrou, então vocês não conseguem mais entrar. E a gente vai sortear de 1 a 22.
Valeu, Bruce. Vai aparecer para vocês em instantes aí. Já apareceu, né? Dormi aqui, gente, desculpa. E agora sorteando número 9. 9 é a pessoa Felipe Bueno. Marmelada. Desculpa. Letícia Garcia. Letícia Garcia levou o sorteio. Parabéns, Letícia. Isso aí, pessoal. Muito obrigado. Aproveita a SEB. Ah, pode falar. Ah, tem um sorteio. Ah, tá. Então o último foi o online, agora é o presencial. Vamos ver. Tem 40 alunos. Então vamos lá, vou colocar aqui o 40. Três, dois, um, 10. Número 10. E aí, Daniela, parabéns. Mais dois? Vai sair o 11 agora, quer ver? 9, 10, 11. 29. Dois patinhos na lagoa. O nosso Breno, o Breno Breno e o último 3. Ana Beatriz, muito bem, pessoal. Muito obrigado, pessoal. Aproveitem mais a SEB, tem mais momentos aí. Até mais.